Hey, it's Katie from Philly Wellness Center. So this is something I wanted to talk about for a long time. And I finally kind of figured out how to put this together. So cholesterol and cancer. So, there's a lot of studies out there and they seem to completely contradict one another. So especially pharmaceutical funded trials, they often conclude that statins in particular and low cholesterol have no impact on cancer risk. Yet we have decades and decades of biochemistry that show something very different. So how is it that these two things can both be true? So cholesterol is it's vital for cellular integrity, immune precision, and even DNA protection. So in this video, we're going to explore two completely different ways of studying the same molecule, one that looks at populations and one that looks at cells. And at the end, you'll understand why the truth depends on which lens you use, and then you can decide which lens you want to use. All right, so the first lens is the pharmaceutical lens, right? So if it asks if we give someone a statin to a large group of people, does the total cancer rate change? That's an outcome-based question. It doesn't measure what's happening inside the cells, it just counts events across the population. The second lens is the biochemical lens, and it asks, well, what happens to the cell membranes, mitochondria, and immune signaling when cholesterol availability changes? That is a mechanistic question, and it reveals the cause and effect at the cellular level. So population studies average outcomes across thousands of people. Biochemistry shows the molecular story beneath the averages. All right, so let's start with what we know from pure biochemistry, work done in cell cultures, animal models, and fundamental metabolism research. First, membranes and lipid rafts. So cholesterol is what gives your cell membrane structure and order. It forms these things called lipid rafts, which are microdomains that organize receptors like insulin, IGF-1, and immune signaling proteins. Now, when cholesterol is low, those rafts start to disintegrate, right? The signals become chaotic, receptors don't cluster properly, and the cell loses communication with the environment. Second, mitochondrial energy, the same pathway that makes cholesterol, also produces CoQ10, which is a vital electron carrier in the mitochondrial respiratory chain. If that pathway is suppressed, you don't just make less cholesterol, you also make less CoQ10. Less CoQ10 means less ATP, more oxidative stress, and more leakage of reactive oxygen species, all which promote DNA damage, which can lead to cancer. Third, lipid peroxidation. When membranes lose cholesterol, they become more polyunsaturated. That makes them more chemically fragile. Reactive oxygen species attack these unsaturated bonds, producing aldehydes like 4-HNE and malondialdehyde, and they bind to DNA and proteins. These are direct mutagens. They can literally initiate carcinogenesis. Okay. Big problem. So lipid peroxidation, huge problem for the cell. So the cell has a very specific way of neutralizing lipid peroxidation that we'll talk about. And fourth, immune surveillance. T cells and natural killer cells rely on cholesterol-rich membranes to form immune synapses and target abnormal cells. So low cholesterol impairs this process, weakening the body's ability to detect and destroy emerging tumors. So the biochemical level, the story is consistent. When cholesterol is low, membranes are weak, mitochondria fail, and immune defenses falter. All of these conditions make cancer more likely to arise or progress because this becomes an opportunistic uh, situation for cancer. All right, so what's the deal? Let's with the pharmaceutical studies. Let's switch lenses for a second and see why large human trials often look neutral, right? So first, reverse causation. So people with undiagnosed cancer often have low cholesterol because tumors consume cholesterol or suppress liver synthesis. So low cholesterol might reflect disease, but not cause it. Second, outcome dilution. When studies combine every cancer type, liver, breast, brain, whatever, blood, opposite effects can cancel each other out. Third, timing. The cholesterol requirements of a healthy person are different from those of a healing or an inflamed person. So lowering cholesterol might not trigger cancer in a healthy adult, but could impair recovery after cellular injury. Fourth, heterogeneity, nutrient status, oxidative stress, hormone balance, and mitochondrial efficiency all influence whether low cholesterol is harmful. Population studies rarely control for these factors. And finally, compartmental mismatch. Serum cholesterol doesn't necessarily reflect cholesterol inside the brain, immune cells, or membranes, where the biochemistry actually happens. So when the pharmaceutical data says that statins don't increase cancers, technically that's correct at the population level, but it doesn't mean that low cholesterol is harmless at the cellular level. So here's how to hold both ideas without contradiction. Population studies tell us that cholesterol-lowering drugs don't create a sudden cancer epidemic. Well, that's good. But mechanistic research tells us that cholesterol homeostasis, not depletion, is essential for resilience, repair, and long-term cellular health. In other words, population data gives us safety averages, but biochemistry tells us what keeps the cell alive. So when you average together 1,000 lifestyles, diets, toxin exposures, and stress levels, the signal disappears, but the underlying biology does not. So instead of asking, is cholesterol good or bad? The better question is, what is happening? What's going on here? In chronic inflammation or oxidative stress, lowering cholesterol can remove one of the body's primary defenses against membrane oxidation, that lipid peroxidation. In recovery from brain injury or stroke, their cells need cholesterol to rebuild myelin and synapses. In immune surveillance, cholesterol-rich membranes help T cells identify and kill precancerous cells. So low cholesterol isn't inherently bad, but in the wrong context, it removes essential building blocks for repair. Large studies that show statins don't increase cancer on average, but biochemistry shows why cholesterol depletion can still be harmful in certain biological states, especially when tissues are trying to recover, repair, or manage oxidative stress. So the key takeaway here is outcomes data sets boundaries, but mechanistic data shows what's happening inside those boundaries. So the next time you hear there's no link between cholesterol and cancer, remember to ask, what are we looking at here? So on a spreadsheet, maybe not, but is that the risk that you're going to take? Is that is that good enough for you? Because inside a cell, cholesterol is life, structure, and energy itself. And if you want true prevention and recovery, you can't just lower numbers. You have to understand the biochemistry and what's keeping those cells alive.